Next, we would like to welcome Mike Blundell from Bio-Rad, and he's going to discuss the Starbrite ultraviolet dyes and sort of continue the discussion that we were just having. So take it away, Mike.
Okay, so the obligatory, can you see my screen?
Yes, we can.
Can, excellent. Okay, so yeah, I'm going to just thank you for inviting me to talk, and I'm just going to talk a little bit more about Starbrite and some of those properties in flow cytometry. So, I'm going to talk about properties, although Daniel's actually explained them very well, show you some of the performance, and then show you some of the sort of higher parameter multicolored data we have, talk a little bit about our future releases, and then just some of the resources and how we can help you. So I have this slide, what are Starbrite dyes? We just had a very nice talk on what they are, but they are unique fluorescent nanoparticles specifically developed for flow cytometry, and they do have that superior brightness with a narrow excitation and emission, and you're able to produce large Stokes shifts without being this traditional tandem dye. We've given them a simple nomenclature of Starbrite and then laser line color, then the emission peak. And I've just got some examples here of the brightness. So we have a Starbrite Blue 700, and it's as bright as Brilliant Blue 700, brighter than Percy Psi 5.5. And then if you look at the stain index right across our Starbrite Violet range, you can see that they are brighter or are as bright as every single one of the Brilliant Violets. They're brighter than the Super Brights here in green, and brighter than things like Pacific Blue and any other dyes out there. So they are very, very bright dyes. So this is what we have on sale at the moment. So we have a full range of Starbrite Violet. We're just missing one actually, but these are the competitor dyes for the, the polymer dyes, and then Bio-Bright dyes, Pacific Blue, Pacific Orange. And we have one Starbrite Blue 700, excitable by the 488 laser, which again is this competitor for Percy Psi 5.5 and Brilliant Blue 700. Now, one of the first things I want to talk about Starbrite dyes is they actually do work in all buffers. You do not need a special buffer to multiplex Starbrite dyes. So we actually do most of our staining in just PBS BSA, but we have our own commercial buffer. But Starbrite dyes do work in Brilliant Stain Buffer if you are using lots of dyes that do require that Brilliant Stain Buffer. We see no change in performance. In fact, you can add sodium azide, you could add EDTA, so if you're doing sorting of sticky cells, for example, again, we see no difference. So all common staining buffers, they get great performance. And we just have a very small example here. So if you have 2-color staining, 2 Alexa dyes, you get CD3, CD8, there's your cytotoxic T cells. You swap in 1 Starbrite dye, no change, and you swap in 2 Starbrite dyes, and you're still getting that same pattern of staining. There is no interactions between these dyes. If you do want to use multiple other polymer dyes, for example, we still say use a special buffer because we can't say that they are not going to interact with each other. You get very, very good reproducibility with the Starbrite dyes. So there's minimal lot-to-lot variance. And here I just have some data where we made 3 lots and we stained the same sample 3 times with those 3 lots. And we looked at the brightness and the stain index, and we just looked at the target population you could identify. And what we can see is the overlay here of all 9 experiments, and this is Starbrite Violet 515 compared to Brilliant Violet 510. You can see there's very little variation. And in fact, if you look at the numbers, the standard deviation is about 10%. So very, very good. And in fact, you're identifying almost the same percent positive every time as well. So you're going to get the same results in your longitudinal experiments. Starbrite dyes, as we just heard, are very, very stable. But when you actually conjugate them to an antibody, they're still stable. So we have an example here of accelerated stability. So we kept the antibody, the conjugated antibody at a higher temperature, and it mimicked up to 51 months at 4 degrees. And you can see there's no change in that stain index, and there's the staining for up to 51 months. And the comparison dye here, Percy Psi 5.5, you do get a drop within that time frame. So that's just accelerated stability. We got real-time stability for about 2.5 years, and we're seeing no drop in performance. But I just wanted to do a little experiment myself where I took some antibody out of the fridge, we made it up into the right concentration in PBS BSA, and we left it out on the bench. So we did this either in the dark, so we put a bit of foil around the tube, or we kept them in the light, just so the normal ambient light. And then we took a sample, so we have either 4 days, 14 days for both, and then we stained the same sample fresh. And we can see the fresh sample, CD14 in this case, with very, very bright staining, and 14 days on the bench in the dark, there's no drop in performance. And in fact, it took 2 weeks for that sample being left out on the bench before we saw a drop in the brightness of the staining. In fact, after 4 days, it was fine. So, you know, if you accidentally leave them out, it should be fine. I wouldn't do it, but your antibodies should be fine. And in fact, you can pre-mix Starbrites. So we pre-mixed just 4 Starbrites here. It's CD19, CD3, CD4, CD8, just to look at some simple lineages in peripheral blood. And we left them in the fridge as a master mix in PBS, BSA, or in fact in Brilliant Buffer, in our own staining buffer, and in fact just in PBS, for up to 28 days, and then compared to a freshly made up master mix. And we saw virtually identical staining. So regardless of the buffer, although we saw a slight drop with PBS, so we would say include a carrier if you want to premix. So this is good. If you've got lots of samples coming in over a few weeks, you can make up a great big master mix and reduce some of your pipetting errors. And Starbrite dyes can be fixed. So we have data here comparing fresh sample and then fixed in our own Bio-Rad fixation buffer, or 2% paraformaldehyde. We have gone up to 4% paraformaldehyde. We've seen no change. And in fact, we have done some preliminary tests using alcohol fixation. And again, we see no drop in performance if you fix either in paraformaldehyde or in alcohol. So what I really want to show you today is that we are about to launch Starbrite ultraviolet dyes. So as Daniel said, we actually have a range spanning from 400 to 795 nanometer emitting. And we have one new one here, Starbrite Ultraviolet 445, for which there is no real competitor polymer dye. So there's 8 new dyes, and these have that same brightness, the narrow excitation emission, the instrument compatibility, work in all buffers still, consistent staining, and suitable for multiplexing. And we just have an example here to show the brightness. So the Starbrite Ultraviolet 400, and the competitor dye here is BUV 395. You can see a lot brighter. I don't have the full set yet to do the stain index, but you can see where we have the dye available, we are brighter than the competitor dye. And so we've used those dyes, combined the ultraviolets and the violet dyes and the Starbrite Blue 700 in an 18 color panel in conventional flow cytometry on the ZE5. So this is 18 color panel with 11 Starbrites in it. And we have other dyes, common dyes, you know, the Alexa dyes, PE, tandems, PE, FITSI, and this staining was done in PBS BSA. And as you'd expect, we have some just some simple lineage determination to identify B cells and T cells, look at CD4, CD8 populations, look at some memory phenotypes using CD57 or CD45 RA and RO, then looking at different effector memory, central memory, naive, terminal effector memory, looking at co-stimulating molecule expressions such as CD28, CD27, Tregs. So we can see here, based on CD127 and CD25 surface expression, we can see really good separation of the Tregs, and we can identify our granulocytes, monocytes. So everything you'd expect, you get really nice separation of these dyes, of the populations using these dyes. And if you want to see the spillover and spreading matrix, this is available, or will be available on our website very soon. In fact, we do have this as a poster from Cyto available as well. And so then we thought, well, okay, you know, everyone doing spectral cytometry now, so let's do a larger panel. So here we did a 27 color panel, and this has 13 Starbrite dyes in it. Now this panel also included some brilliant ultraviolets and some brilliant violet dyes. So this was done in Brilliant Buffer. But you know, with the game, we have the Alexa dyes and tandems, all the common dyes you'd expect. And because we had more colors, we were able to identify more cell populations. So NKT cells, gamma deltas, NK cells, different granulocyte populations. We could identify everything you'd expect and still see all the T cell populations, different memory, effector memory populations, etc. What I want to point out really here is we've got some novel combinations with the Starbrite dyes because they do have unique spectral characteristics, unique spectral profiles. You can combine them with the brilliant violet dyes and with other dyes. So we have a combination of BV421, Pacific Blue, and Starbrite Violet 440 together. We had Starbrite Violet 475, BV510, and Starbrite Violet 515. BV605 with 610. And we put Starbrite Violet 710 with 711, and Starbrite Blue 700 can be used with PerCP Sci 5.5. So you can see here, there's some novel combinations that you wouldn't be able to do previously. So looking at this, we actually have the similarity score and where we think the dyes are compatible. I've marked it. So for the Starbrite Violets, we know that most of these are compatible. We're unsure at the moment about Starbrite Violet 475 with BV480 and Starbrite Violet 570 with Brilliant Violet 570. And we know that most of the Starbrite Ultraviolets are actually compatible with the Brilliant Ultraviolet series. The first one we're bringing out, unfortunately, is not, because the similarity is too close, and one of them we don't yet know. And Starbrite Blue 700 compatible actually with Brilliant Blue 700 and PerCP Sci 5.5. So we think there's at least 12 novel combinations, although some of them do have high similarity scores. So we would say, you might get a bit of spreading. So maybe if you want to add these dyes together, you could put them on mutually exclusive markers. And in fact, that's what we did in that 27 color panel. And I think really it shows the potential of these dyes. You can probably get over 50 colors now, quite straightforward, by combining the Starbrites with other say violet and ultraviolet polymer-based dyes. But we're not stopping there. First off, so these are the available antibodies we have. So we have some human targets and some mouse targets. We've gone with common immunophenotyping targets first off, and we have streptavidin. So these, you notice these are cell surface targets only. We are working on intracellular targets. We want to get a protocol that is right to make sure that you don't have to troubleshoot, and we can do the troubleshooting for you. But what we do know is the Starbrite dyes work with fixed perm buffers with minimal drop in performance as well. So if you are doing fixed perm and looking for cytokines, for example, you do your surface staining first, the Starbrites will still be fine afterwards. As I say, we're not stopping there. We've got the Starbrite Blue dyes. And this is to expand the number of colors, obviously, on the 488 laser. We've tested these in spectral, and they're bright with unique spectra, and they're compatible with other 488 excitable dyes and with PE tandems. And we've got a whole series here, and I just got some example data. We're not stopping there. We're actually going to start on the Starbrite Yellow. So these are going to be 561 excitable, same bright and stable as the rest of Starbrite dyes. But here, we have less excitation by the 488 laser. So they should be more compatible with blue excitable dyes. And we just have one example to show you here. So our Starbrite Yellow 665, when combined with Starbrite Blue 675, we see the staining, and the comparison staining with PE Sci 5 here, the spreading is a lot higher. Admittedly, it's a bit brighter. So just an overview, they're brighter than competitor dyes. We have narrow excitation and emission. They're compatible on all instrumentation with the right lasers and filters, including spectral. They work in all buffers and don't need a special buffer to multiplex them. You get very consistent staining, and you can fix with PFA and alcohol, and you can pre-mix them. And of course, this just increases your multiplexing capabilities. And we have them available on common immunophenotyping targets that are known clones and highly cited. We have some support and resources for you. So we have dedicated web pages, and we have online tools such as a spectra viewer. We have some panel data and the excitation and emission profiles. And then in addition to that, we have some general flow cytometry information, so flow guides and cell frequency antigen density to help you build those panels better. The spectra viewer, just want to point out, actually opens out to show you the excitation and emission by each laser. So you can see that cross-laser excitation and emission. It doesn't just contain Bio-Rad fluorescent dyes, it contains all of them, including fluorescent proteins. It's customizable, and it's mobile device compatible as well. and it appears on the product pages. So you don't have to leave the product page if you're going to be looking at certain products. So if you want to find out more, you can sign up to receive our emails. You can find out more about Starbrite dyes on bioradantibodies.com/Starbrite. Or you can contact your local representatives. So, thank you.
Okay, thanks for a great talk, Mike. It was very informative. There are a few questions in the chat. I'll just get to one very quickly that is Starbrite specific. Amina asks, how well do Starbrite conjugated antibodies work on whole blood and whole blood collected in Cytochex tubes?
Well, all of our staining is done on whole blood. We stain and then lyse afterwards. So it's fine. I don't know about the Cytochex too. I'd have to find out.
Okay, great, thanks.